everybody online. Now it's 3 p.m. Let's start with the talk. So we are glad to see you online for this web seminar. So today we're gonna have the speaker from Donghua University. So I'm very be honored to be the host of today's group seminar, the seminar. So I'm from the School of Pharmacy in Guangzhou Medical University. I received my PhD degree in the Macquarie University, and after that, I was trained my postdoc in the University of Minnesota at VCU. Currently, my research topics are mainly about the lipid nanoparticles for the delivery of therapeutics and vaccines. Nice to meet you, and welcome to collaborate with me if you have any opportunities. So let me briefly introduce this journal is Biomedical Engineering Horizon is BMEH, and is very good journal. It's an open access journal, but however, it's free of charge until for the papers accepted before the end of 2029, and the scope is very is broad in from bacteria, from biomaterials to bioinformatics, computational biology, or nanomedicine. Firstly, the editor-in-chief of this journal is Professor Ma, and the junior executive editors is Professor Xue and Xu. The associate editors are Professor Chen, Dean, Professor Zhang, Professor Wang, Lin Kelly, and Professor Lammers. And we also have a group of editorial board members and the youth editorial board. Now we will welcome the Professor Shi to introduce his talk titled in Fibronectin-Based Nanomedicine Design and Applications. Professor Shi is the professor in and the Vice Dean of the College of Biological Science and Medical Engineering in Donghua University, Shanghai. And he became the professor of special appointment both in Donghua and Shanghai Institution of High Learning, and he is the Eastern Scholar since 2008 and he is also he serves as editor in different scientific journals such as the Bioconjugates, Chemistry, and Nanomedicine. And his research interests are focused on the development of organic or inorganic hybrid nanoplatforms or macrofluid platforms for sensory image and diagnostic application. So now, welcome Professor Shi for his talking.
Okay. Let me see.
Okay, we will stop the sharing and give it to you.
Okay. It's still here, yeah. Can you stop the sharing?
Yeah, but I'm trying. I know. Okay.
All right.
I found the button.
Okay. Okay, just a minute. Um, so can you see my screen?
Yes, it's very clear.
Okay.
Yeah,
All
we
right.
can start.
Okay. Yeah. First of all, it's really my good pleasure to be invited to have a talk. So it's for the BME Horizon. I think it should be like a very nice journal. Uh, so we're looking forward to contribute to this journal. And today, I'm going to talk about my recent work regarding fibronectin-based nanomedicine design and applications. And here is my uh, outline of my talk. I will briefly introduce some of the background, then mainly talk about how we design polyamidoamine dendrimer for fibronectin delivery. Then we'll talk about how we design phosphorus dendrimer for the delivery of fibronectin. And lastly, will focus on we are going to use fibronectin delivery to tackle some brain diseases. And if you look at the market, the best-selling pharmaceuticals globally in the year 2023, in the top 10 pharmaceuticals, the top 10 among the top 10, there are 7 drugs are protein drugs. So this means like a protein drug has been like widely adapted and used in the industry. And why proteins? Because protein can be used as a carrier or um, or as a therapeutic agents for drug delivery, immunotherapy, as well as disease diagnosis. So the market is keep going on. And among the different uh, protein drugs, so we are concerned to focus on the fibronectin because fibronectin is a kind of uh, emerging protein drugs. If you look at the market globally, the market size surged in 2022 with a project CAGR through like 2028. So according to the current market, there are several main suppliers of fibronectin. One of the main suppliers is from like Shanghai Xianlian Biotechnology Cooperation, and also they are from Suzhou or some other area. So we have been long collaboration with Shanghai Xianlian Biotechnology. So um, originally, Shanghai Xianlian, they are focusing on the cosmetics. So now, we are doing collaboration, trying to develop like novel drugs for different therapeutic applications. And if you look at fibronectin is a high molecular weight glycoprotein. They have 220 kD subunits. Each has 6 dense global domains. So fibronectin has been widely used as a wound repair, tissue regeneration, cancer diagnosis, as well as antibacterial or skin care. So mainly if you look at the structure, uh, it's like a glycoprotein with a multifunctional domain. It contains the RGD peptide sequence. So this sequence is very important in promoting the self-assembly process of the materials and also have a good targeting specificity to target different cells that have overexpression of alpha-V beta-3 integrins. So for fibronectin biomaterial interface, usually fibronectin can be used to coat on the nanoparticle surface for targeting purpose and can be doped into biological scaffold materials to promote cell growth, angiogenesis, and wound repair. And also, you can use fibronectin for anti-inflammatory material surface modification to induce macrophage M2 polarization through the integrin receptor mediated interactions. So the advantage of the fibronectin protein drug is obvious. They have very high activity, have high specificity, low toxicity, good bioavailability, as well as they have a clear biological functions conducive to clinical applications. But the application of fibronectin can be limited because they have poor cell membrane permeability. And why it's important to let the fibronectin to transfer the inside the cell, because fibronectin is a signal protein. It's not just like a scaffold protein. So therefore, it's very important to deliver fibronectin into cells to target intracellular pathways. So in this case, the therapeutic efficacy can be further enhanced. So therefore, it's vital to develop intracellular delivery technology for FN. So in terms of the protein intracellular delivery approach, there are several different methods. First of all, method is the physical methods like electroporation or microfluidics. The advantage is like they have a strong versatility, high efficiency, but the disadvantage is like rely really on the stringent equipment and conditions. And the second method is protein modification. For example, you can modify with cation polymers or cell-penetrating peptides. The advantage is like protein can cross the membrane inherently, but the disadvantage is like you have to have use the covalent conjugation reaction that will impact the protein activity and stability. So therefore, the third methodology is like a nanotechnology. So in this case, the protein can be coated or interact with different nanoparticles through non-covalent interactions. So in this case, you can enhance the membrane permeability as well as stability without disrupting the protein structure. So among the many different nanoparticles, we are concerned to use like dendrimers, especially polyamidoamine dendrimer, because this dendrimer has been commercially available in Dendritech as well as in Weihai Chenyuan in China. So they have multi-functionalization, they have internal cavity and have high loading capacity. So in our earlier work, we're using polyamidoamine dendrimer as a carrier. We can load contrast agent. We can load drugs through covalent or non-covalent way. We can also load it with different biomolecules like proteins or genes for different delivery applications. And in another case, we are also concerned to use phosphodendrimers because phosphodendrimer, which is pioneered by JMP Maharo from CIS, the laboratory coordination chemistry. And the main advantage of phosphodendrimer, they have uniform molecular weight. They have no structural diversity. They have a rigid structure. They have a good biocompatibility, and they have inherent bioactivity. So in earlier work, people use phosphodendrimer for gene delivery and for incorporation with metal ions for intrinsic anti-tumor activity. And also, if the phosphodendrimer has been modified with sodium phosphide salt, they have intrinsic anti-inflammatory activity as well as immunomodulatory activity. So in the past several years, our group has been really concentrate how we develop like fibronectin fibronectin nanomedicine for cancer or for different inflammatory disease treatment. Like for example, for cancer targeting, for acute lung injury relief, and for like arthritis, for osteoarthritis, as well as for neurodegenerative disease like Parkinson's disease, like the ischemic stroke therapy. So in the first example, it's like we are just introducing how we develop like dendrimer based delivery vehicle for fibronectin. And since fibronectin, this protein drug has been used like in the COVID-19, especially in Wuhan, and they have some clinical cases like around the treatment for the COVID-19, they are like 75 to 80% successful rate. So the most of the like old people more than 80 years old, they get survived. But at that time, there's no like animal studies, and there's no too much evidence to show the effectiveness of fibronectin for acute lung injury. So here we are using generation 5 PMM dendrimer we modified with phenylboronic acid. And then like this, the PBA group has a nice binding affinity to protein, so we can form the nanocomplex. So this nanocomplex can be easily delivered to macrophage cells. So in this case, we can tackle the reactive oxygen species in the macrophage. We can also switch the macrophage to anti-inflammatory M2 phenotype. And here is the results. It's very easy. We just use like our dendrimer modified with PBA mixed with fibronectin, and the optimal mass ratio around 8:1. We can form very nice complex with uniform morphology, and we show that the fibronectin can be taken up by cancer cells. So this is the first study. We show that okay, the fibronectin without dendrimer complexation cannot cross the cell membrane. But if you complex with dendrimer, you form the nanoparticle. The nanoparticle can be taken up by cancer cells. Then we trying to switch to macrophage cells, and we show that our dendrimer fibronectin complex can be taken up by macrophage cells, and can switch the macrophage to M2 phenotype. And also you can see this can be you can see the significant induction of M2 macrophage polarization after 48-hour phagocytosis. And we build up kind of a mouse model of the acute lung injury, and we establish this model. We are using nebulization at the bronchial level for in vivo evaluation, and we show that after the treatment, you can see the injury of the lung has been decreased, and the ROS, the ROS, has been scavenged. And you can see like there are significant downregulation of primary inflammatory factors. So just a very short conclusion is like we are using a PBA modified generation 5 dendrimer. So this dendrimer can form we can form with fibronectin to form like a uniform nanocomplex, and the optimized mass ratio for efficient intracellular delivery of fibronectin. And this delivery, once you deliver to macrophage cells, you can switch you can repolarize macrophage to M2 phenotype, can reduce the ROS level. And also we show that can significantly inhibit the NF-kappa B pathway, thereby suppressing the macrophage inflammation. And overall, the nanocomplex can alleviate the acute lung injury symptom in mice by reducing the primary inflammation and oxidative stress, showing super therapeutic effects than the free fibronectin. So, like since like a polyamidoamine dendrimer like has no intrinsic bioactivity, and for like to form the complex, you have to modify PMM dendrimer with kind of aromatic ligand. So this ligand can help to interact with protein to form the stable nanocomplex. So therefore, we are trying to switch to phosphorus dendrimers. The phosphorus dendrimer, they always have like aromatic backbones, and here we are trying to develop like a polyanionic phosphate terminated phosphorus dendrimer. as a universal carrier for protein delivery platform. And why is a phosphide terminated phosphodendrimer? Because this phosphide terminated dendrimer, they have intrinsic immunomodulatory capability, namely anti-inflammation activity. They are an anionic delivery vehicle, which is different. In most cases, like for protein delivery, the vehicle, the common vehicle is always positively charged. But in our case, we are using negatively charged dendrimer as a vehicle to load different type of proteins. So the method is simple, just like mix and centrifuge and purify, you can form the nice complex. You can synergize the immunomodulatory effect in two different inflammation models. So here we are trying to first of all, we are working with a BSA because the BSA is a model protein. So we show that after optimization, you can form a uniform spherical nanoparticle. And this through AFM, you can see the height of the nanoparticle is around 23 nanometer, and the BSA phosphodendrimer nanocompound has good color stability and can be taken up by cells and through the clustering mediated endocytosis pathway. And with this, we also with the optimized condition, we use the phosphide terminate dendrimer, phosphodendrimer to complex fibronectin, and in this case, we cultured with macrophage cells. We cultured with macrophage cells, and we show that the M2 macrophage can be induced, and also you can scavenge the RS to enhance the fibronectin antioxidant activity. So overall, fibronectin itself has intrinsic antioxidant activity as well as anti-inflammation activity. So in this case, the materials, the carriers, the bioactive carrier complex with the bioactive protein. So in that case, the bioactive rise for better treatment. And we show that after the FN delivery to the macrophage cells, the mRNA expression level of the pro-inflammatory cytokine decreased, and the PI3K NF-kappa B signal pathway are inhibited. Okay, so we also built up the same mouse acute lung injury model, and we show that after the treatment through nebulization, can quell the lung overactivated cytokine storm, can inhibit the neutrophil invasion, can improve the primary edema, reduce the lung damage. And we also built up another inflammation model to see if it's like a universal. And for anti-inflammation therapy, where we also built up acute gout arthritis model, and we show that after the injection, the materials can prevent the ankle swelling of the HA mice. So you can see the joint cavity has been normalized. The synovial area in the synovial, the inflammation cell infiltration was significantly inhibited. So this means like the dendrimer, fibronectin, the carrier, and the protein can form the therapeutic complex, can synergize the activity of the dendrimer as well as FN to relieve the inflammation of different inflammatory disease. So just a brief conclusion of this part of the work. So we have synthesized like 5 types of phosphodendrimers. We just find one with the optimized anti-inflammatory and protein delivery properties that has been used to complex different proteins like BSA, the ribonuclease A, or ovalbumin or fibronectin. We just use fibronectin to showcase like we can use fibronectin for nanomedicine applications to tackle two different inflammation models like AGA as well as ALI. Okay, and we show that the developed like polyanionic phosphide terminated phosphodendrimers as a universal carrier system for intracellular protein delivery. So furthermore, like we saw that not only the dendrimer can just complex with the protein, so we can also show like dendrons, like dendrimers. Once you combine with different a nanocarrier system, you can also deal with different inflammatory diseases. Here, like we are using G4-TBP dendrimers. This dendrimer do not have water solubility. So in this case, we are develop like a PEG, the TKPLGA micelles. So these micelles can self-assembly to form the micelles that can encapsulate with the G4-TBP, and the whole micelles can interact with fibronectin to form the nanocomplex. So here, the fibronectin is on the outside of the nanocomplex. It can target the inflammatory microenvironment because the fibronectin has RGD sequence. And the whole materials can scavenge ROS, can attenuate the hypoxia, and can have like M2 macrophage polarization. And also for the bone mesenchymal stem cells, because of the fibronectin, these stem cells can be recreating to the inflammation site, can also promote the osteogenic differentiation and promote the bone repair and osseointegration. So here you can see the nanoparticle has uniform size and can be complexed with fibronectin. They have a uniform spherical morphology, and through different like AFM as well as UV spectroscopy and this potential hydrodynamic size, we show that we have successful synthesized this nanocomplex. And we saw that because of the RGD sequence on the fibronectin, the whole nanocomplex have a targeting specificity to macrophages to enhance the cellular uptake. And once it's taken up by macrophages, they have decreased ROS and lipid peroxidation levels, and can enhance the catalyst enzyme activity to have oxygen generation. So in this case, can relieve the hypoxia inducing factor 1 alpha, the HIFA downregulation. So in this case, can better promote the anti-inflammation and repair for the osteogenesis, osteoarthritis therapy. And through the in vitro work, we suggestively enhance ability by increase the ARG1 and decrease loss to polarize the macrophage to M2 phenotype, can help downregulate pro-inflammatory cytokines, can decrease the cell apoptosis, okay, to protect it from the LPS-induced oxidative stress and inflammation. And we also show that our materials can promote the differentiation of stem cells. You can see the gene expression of the BMP2 and GPMB in macrophage has been enhanced. And also you show the gene expression of the RONX2 and OCN in the stem cells, okay? And the stem cells can go through the osteogenic differentiation, like you can see the activities of the ALP as well as the alizarin red staining to show that our materials promote the osteogenic differentiation of stem cells by immunomodulating the macrophages. And we also built up in vivo mouse model, and we show that after the treatment, the materials can boost the cartridge proteoglycan and alkyl expression, shielding cartridge from damage and degradation. And you can see also the synovitis relief. You see the minimal inflammation in the knee, synovial and infrapatellar pad tissues. And you can also show the significant reduction of the pro-inflammatory factors like TNF-alpha, IL-1 beta, as well as MMP13. And through the histological examination, as well as imaging application, imaging measurement, we show the clearance of the ROS in the knee joint region, the reduction of the HIFA, elevate oxidative stress and hypoxia in vivo, increased number of CD146, BMP2, and OCN positive cells. So overall, just a brief conclusion of this part. So we are developing mast cells encapsulated with phosphodendrimers that can be further modified or formed with fibronectin to form the complex. So this nanocomplex can release the dendrimer as well as fibronectin responsively to the ROS and specifically target macrophage where FN-mediated targeting can enhance the BMSC recruitment and osteogenesis, can curb the cartridge degradation and aid cartridge repair via ROS clearance, the hypoxia elevation, M2 polarization, anti-apoptosis, as well as the BIM, the optimization for arthritis treatment. So, in the first part, I talk about the polyamidoamine dendrimer. In the second part, I will talk about the phosphodendrimer. And actually, this type of protein for therapeutics, we can deal with different inflammatory diseases. Like for example, a neurodegenerative disease like Parkinson's disease, Alzheimer's disease, as well as ischemic stroke. They are all kind of neuroinflammation disease models. So here we are trying to deliver fibronectin through the phosphodendrimers to bring for Parkinson's disease treatment via cooperative modulation of the microglia. So here we are using AK123 dendrimer with hydroxyl terminal group. We can just quickly or easily form with fibronectin to form the nanocomplex. So why hydroxyl terminated phosphodendrimer? Because this hydroxyl group can allow the complex to cross the blood-brain barrier. And also through the FN mediated targeting, because of the RGD sequence, you can target the inflammatory the cell types like microglia, or as the damaged neurons. And through the synergistic bioactivity integration of fibronectin and the phosphorus dendrimers, you can cooperatively modulate the macroglia for macroglia M2 phenotype polarization, for ROS scavenging, for oxidative stress and inflammation inhibition. So here our the design is very simple, just two materials, very simple composition. And see once you optimize the mass ratio between fibronectin and the phosphodendrimers, you can form uniform nanoparticles with spherical shape. And you can also see through the CD spectra, the secondary conformation of the fibronectin has been retained. And the overall nanocomplex has desired colloidal stability in water, PBS buffer, as well as cell culture medium. And through the SDS page measurement, we show the successful loading of fibronectin, and the overall the materials have a good cytocompatibility. We also show that the fibronectin phosphodendrimer nanocomplex has no immunogenicity because we cultured with dendritic cells to see the maturation status, and we show that the nanocomplex possess non-immunogenicity. And through the in vitro assays, and we show that our nanocomplex can modulate microglia and for M2 phenotype polarization, can scavenge the ROS in the macrophages. And through the Western blot assay, we show that the materials can inhibit the NF-kappa B pathway, can decrease the pro-inflammatory factor secretion. And we also built up an in vitro transfer BBB model, and we show that our nanocomplex can cross the BBB, and they have a nice hemocompatibility. And in vivo, we show that the materials, once you do intravenous injection, the materials can cross the BBB to show a nice signal, fluorescent signal in the brain. And we also show that through the pharmacokinetic studies, the fibronectin blood circulation half-life can be extended. Okay, and we did like a behavior test, and then we showed like from the rotary grip, suspension, forced swimming, and also like you can see some of the results. Like the tail suspension, forced swimming, as well as open field. And you can see that after the treatment with like our nanocomplex can improve the depressive, anxious behavior of the PD mice. Okay. And we did the histological examination, and we show that through HG staining of the hippocampus, you show that the nanocomplex can ameliorate the cellular condition of the hippocampus in mice, restoring the nice body content, can decrease the IBA1 expression, increase the TH expression, and elevate the dopamine level in the brain. And also, since like alpha synuclein protein is the marker of the Parkinson's disease, we show that the level of the alpha synuclein as well as GFAP can be decreased after treatment, which is quite close to the healthy control. And we also show that after the treatment, it can increase the CD206, CD86 level in the brain, which means that the microglia phenotype has been switched to M2 phenotype. And we show that after treatment, the materials, the overall materials have no cytotoxicity, has a very nice biosafety profile, and there are no long-term toxicity because the phosphodendrimer and fibronectin, they are both biocompatible materials. So in this case, I just give a brief conclusion, like the developed nanomedicine based on hydroxyl-terminated bioactive phosphodendrimers compounds with FN have a nice water dispersion stability and cytocompatibility, and can target to microglia and cross the BBB and regulate the microglia polarization, and show remarkable anti-inflammatory activity. So the whole materials can penetrate the damaged BBB and exert the anti-inflammatory and antioxidant effect, resulting in effective elevation of the PD, the behavioral symptoms of PD mice. So in the last example, I'm going to talk about, like how we use phosphodendrimer to co-deliver fibronectin and interleukin for combined ischemic stroke treatment. See, like since like ischemic stroke is like accounting for like more than 80% of the stroke in the patients. So like here we're using like phosphodendrimer with phosphide terminal group complex with fibronectin to form the nanocomplex. This nanocomplex has hydrophobic internal domain. So this domain can be encapsulated with eutrophin, which is also hydrophobic. And then like we are using macrophage membrane through the extrusion method, we can coat the macrophage cell membrane on the surface of nanocomplex. So here we are using like AK 137. This can influence the melanocytes for anti-inflammatory treatment. The FN have anti-inflammation as well as antioxidation effect, and eutrophin is a free radical scavenger drug. And you can see like we can form like very uniform nanocomplex. Like the Z potential is around 9 minus 9.5, which is close to the macrophage cell membrane. And we also show through the SDS page to show like the fibronectin has been successfully loaded and the cell membrane has been successfully camouflaged. And we show that the materials has a nice hemocompatibility. And you can see here, after cell membrane coating, the thickness of the cell membrane are very clear. And the drug can be responsively released. At acid pH, the drug can be fast released. And the overall, the materials has nice colloidal stability in different aqueous medium. And we show that the overall materials are cytocompatible to the microglia cells as well as to neuron cells, but they are also have nice cellular uptake efficacy because of the macrophage cell membrane coating to both macrophage cells as well as the neuron cells. And we saw that after the treatment of the microglia, you can see the RS can be scavenged, and also the materials can cross the in vitro BBB model and can polarize BB2 cells to M2 phenotype, can decrease the inflammatory factors. And also you can see like for even for neuron cells, we have a similar effect to scavenge the RS and to like the mitochondria membrane potential return to normal level. Also, the mitochondria of the neuron cells, the morphology can be recovered back to the normal level. Okay, and we show that because fibronectin has angiogenesis activity, we show that the whole complex can also have the angiogenetic ability. You can see the tubule formation, which means like you have the blood vessel. This means like after the treatment, the damaged blood-brain barrier can be recovered back to normal. And we saw that after the treatment, the infection area has been significantly decreased through MRI imaging as well as through the TTC staining. And the Z-Langor score, the measurement, we show that around 80% of the Timaco rats could walk normally after the treatment. And also the rotor rod and the four-pole tension test, we show that we can restore the rats fatigue resistance and grip strength levels. And we try to show like through the in vivo experiment, we show that the materials can promote BV to M2 phenotype, can have like the antioxidant protein can be upregulated, and the apoptosis related protein can be downregulated, and can protect the neuron cells in the ischemic brain. And we also did we saw that the blood flow can be recovered to 75% after 4 hours, and can increase the vascular intracellular cells. And you can also see the CD31 has been upregulated. So this means that after the treatment, the vascular regeneration can be improved to like improve the vascular recanalization. So just give a brief conclusion of this part. So we developed like a phosphodendrimer fibronectin nanocomplex. This nanocomplex can be further used as a carrier to load free radical scavenging drug, either and after the macrophages membrane coating, the materials can cross the BBB, can target microglia neurons and endocellular cells for efficient anti-inflammatory, antioxidant, anti-apoptotic, and pro-angiogenic effects for effective ischemic stroke treatment. So just to give a final summary, so we are using dendrimers to deliver fibronectin not only through primary dendrimer but also through the phosphodendrimer. Since phosphodendrimer has intrinsic bioactivity, so we can develop a different drug for different combination to tackle different inflammatory diseases like acute lung injury, like the arthritis, as well as for neurodegenerative diseases. Finally, I would like to acknowledge my colleagues, group members, as well as international collaborators. I think one of the collaborators here, Dimitri Shabin, is here also in his audience today. Thank you very much for your attention. I would like to take some questions if you have. Thank you.